Epimecium. Okay, epimecium is just not on the slide. There's no way to know where epimecium is. Something like this between individual fibers is probably endomecium. Okay, these nuclei, let's make it simple, let's make a diagram of it simple. Okay, these nuclei are either nuclei of fibroblasts making the endomecium, okay, bless you, or they're nuclei of the skeletal muscle cells, or they're stem cells, satellite cells in the skeletal muscle. And it's really kind of difficult to know at a histological level what's what. All right, so I won't really ask you anything about the nuclei like that if it's an image laid out like this. Okay, I may ask what could these nuclei could represent what potential cell type. Okay, and I may have a whole bunch of choices. Okay, with only one of the possible choices as a right answer. Make sense? But I wouldn't ask you, just blind to say what type of nucleus is it. Capillary or a small blood vessel, probably a little bit larger than a capillary, but not much larger than a little arteriole or venule. Skeletal muscle. Again, you can get a sense of the myofibrils, each one of these being many, many myofilament molecules, and you can get a sense, when you see them in cross-section as well. And nice central peripheral nuclei, and looking like this around each of the individual cells. Once again, you would be defining endomecium. Same thing, just skeletal muscle. Okay. Same thing, just skeletal muscle. Okay, maybe we're getting a sense of red and white fibers, or maybe intermediate fibers. I won't ask you to make that distinction on a histological slide, but it does show the difference in staining color is probably reflecting the red and white muscle fibers. And remember, if I say muscle fiber, muscle cell. Not the best micrograph, because it's kind of almost very refractile and maybe artificially separated. But I think there's no question you can say these areas would be endomecium, and maybe this is perimecium around it. Again, I don't know where epimecium is in relation to that. Even here, I don't know with absolute certainty where the edge of the section is. You might imagine if you had some of the connective tissue here or here, you might be on epimecium, but there's not enough information on that slide. No, that. Smooth muscle. Okay, all of these slides are projected at about the same magnification. Okay, so let's go back for a minute. This skeletal muscle, and the nuclei are kind of on the edges of the fibers. Here you can see nuclei, and the trick with it is to say, well, I think I'm seeing a fiber like this, a cell boundary like this. Okay, and if you trace these, in many cases you can see cell boundaries. And it looks like the nuclei are not at the edges of the boundaries. Okay, it looks like they're more in they've got cytoplasm all around them. So that kind of suggests that it's smooth muscle. The other thing, which is kind of a finer point to suggest there's smooth muscle, but it makes a lot of sense from the metabolic activity, is to look at the nuclei. They have like a punctate or a granular appearance, which is seeing maybe little nucleoli, or just clumps of heterochromatin and euchromatin, implying a metabolically active cell. And smooth muscle is very active metabolically, very active synthetically. Okay, so I use that sometimes as a hint if I'm not absolutely sure of that it's smooth muscle. And I also look at them and sometimes see the nuclei look a little bit scrunchy. And remember, when smooth muscle contracts, in the same cell, maybe only part of the cell contracts, and other parts don't. And that will tend to give the nucleus that ruffly or scrunchy type appearance. Whereas in striated muscle, when the cell contracts, the whole cell is going to be contracted. This is smooth muscle. I don't know exactly where it's from. It's probably an organ like the bladder. The muscles run in a lot of different layers, coming, crisscross in and out of the plane of the screen, and almost like layers of plywood that intermingle. And that's very characteristic of an organ like the bladder. This is smooth muscle. On the very top is mesothelium. Because the very top is probably the serosal surface of an organ like the GI tract. I'm just making that assumption because there's a nice layering of muscle running at right angles to each other. If you think of layering in the GI tract, the muscularis externa is an inner circular layer of muscle that's running around the lumen, and the outer longitudinal is running parallel to the lumen. So if the organ was cut in cross-section, that's what you'd see. All right, but once again, you can see the scrunchiness of the nuclei. You can see nucleoli suggesting the cells are metabolically active. Okay, and the other thing that I like to look at in cross-section, you can see the nuclei clearly in the middle, where the nucleus is in the plane of section. And in smooth muscle, the nucleus-cytoplasmic ratio is high. Okay, that's a useful point. Because in cardiac muscle, I'll come back to these, folks stay here. In cardiac muscle, the nucleus is in the center, but the nucleus-cytoplasmic ratio is much lower than in smooth muscle. Okay, you can see the evidence of the myofibrils in the cardiac muscle because it is striated. And you can clearly see the fibers are branched. Okay, you look at this impression that the fibers are branched. Very, very characteristic of cardiac muscle, with the branched fibers. Okay, nuclei are in the center, and I think you can sense that they have striations. And even at this power, you can sometimes see profiles of the intercalated discs, which you can see there too. These are the intercalated discs. Make sense? It's cardiac muscle. It's obviously in the heart somewhere. Where you see that much cardiac muscle. The slide isn't in the great focus, but this would be in longitudinal section and in cross section. Higher power view of cardiac muscle. The intercalated discs are nicely seen. Okay. Very characteristic of cardiac muscle is this perinuclear area, which in histological section often seems clear. Okay, that's where there's a lot of mitochondria and glycogen granules because these have a very high metabolic demand. This one is also interesting. I don't think I made the point of this one in lecture, but this is one cell down at the bottom of the slide. And the cell has two nuclei. Cardiac muscle cells can be binucleated. Okay? To my knowledge, they're not polyploid, but they can sometimes have two nuclei in them. We'll just saw that when we get the central nuclei. This one is a really horrible slide. Okay, it's skeletal muscle. Dr. Scalco has an interesting story on this one. And I don't know if the story is true. Okay, right here is a muscle spindle with the little intrafusal fibers. Those are remember the muscle spindles are the sensory organs in skeletal muscle. These guys are the larger extrafusal or typical skeletal muscle fibers. Okay, those intrafusal fibers in the spindle are the sensory fibers that detect stretch. Okay, Dr. Scalco's interesting story is this came from someone that was electrocuted. Okay, and the interesting story is it was someone that was executed probably in the 1950s, and they donated their remains to science. I don't know if that's true. I don't know if that's why the tissue looks so horrible, but he insists that it's true. Okay, and he's probably right. This is skeletal muscle, and this is the neuromuscular junctions. And I don't know, there may be one that's one higher power with the terminal boutons on them. Okay, now in the glass slides, you're going to fret, and most of you won't see these. Maybe, you know, of 72 slide sets, maybe 10 slides are going to show these. So when you look at these in the lab, look on the QCOM atlas. Skeletal muscle and a tendon. Okay, now this one is tricky because you don't really see the striations. Now, if you're on a microscope, you could focus and see the striations, but you can see the peripheral nuclei, and muscle stains much more eosinophilic, much redder in color, or more orange in color than the connective tissue of the tendon. And the nuclei that you see in the connective tissue are nuclei of fibrocytes, or fibroblasts. And that's the end of muscle.